Assalamu alaikum. Hope you're all well and doing amazing. The video on the topic energy and enzymes or life drivings and controlling forces contains instructive and educational content that is both easy and shareable to retain. So let's start. Here we have the contents that help us to understand the functioning of the enzymes and the structure that helps the enzymes to work very efficiently. So first have a look about the enzymes. What are enzymes? Enzymes are the three-dimensional biological catalysts. They are the globular proteins catalysis biochemical reactions. Some RNA species also act as enzymes called the ribozymes, for example, the hammerhead ribozymes. Now I would like to talk about the characteristics of the enzymes. They speed up the reaction by lowering the activation energy. They are specific in their action and that only one enzyme can catalyze one substrate at one time. They are sensitive to change in pH, temperature, and substrate concentrations. The number of substrate molecule transformed per minute by one enzyme is called the turnover. For example, the catalase turnover number is 6 into 106 per minute. We have the summary of the characteristics of the enzymes like they are highly specific, extremely efficient, stable in structure, required for activators or coenzymes, optimum temperature, dependent on pH and repressed by inhibitors, beneficial enzymes amounts increases, and the last one is function reversely. The next one is about the structure of enzymes. The active site of an enzyme has a specific shape. It binds substrate, prostatic group, cofactors, and residue that hold the substrate. A change in shape of protein affects the functioning of an enzyme. Active sites, there are two types of active sites, binding sites and the catalytic sites. What is binding sites? It selects the substrate and binds it to the active sites, while the catalytic sites, it helps in performance of catalytic action of enzyme. Here in the image, we have very clear the enzymes, active sites, enzyme, substrate, and products of reactions as a result of the reaction. Now, how do enzymes work? They are the biological molecules that speed up all reactions taking place within the cells. They are vital for life and serve a variety of functions such as aiding in digestion and metabolism. They break up large molecules into smaller pieces that are absorbed by the body cells. The molecules that an enzyme works with are called the substrate. The substrate binds to a region on the enzymes called the active site. So enzymes plays a very major role in our lives, and without enzymes, our life isn't possible. In the image, we can see that the enzymes and the substrate forming the enzyme substrate complex and enzymes product complex, and then we have the free enzymes and the products. The next one is about a lock and key model. It was proposed by Emil Fischer in 1894. According to this model, the active sites of an enzyme rigid in shape, no shape in change or no change in shape before and after the chemical reaction. For example, a specific key can open a specific lock. In a very similar manner, a specific substrate can bind to the enzymes forming the enzyme substrate complex. The next one is about the induced fit model. It was proposed by Daniel Koshland in 1958. According to this model, exposure of an enzyme to substrate causes a change in enzyme which causes the active sites to change in its shape to allow substrate to bind. Next one is about the cofactors and coenzymes. Cofactors are the non-protein molecules which carry out the chemical reactions that couldn't be performed by standard amino acids. There are two types of cofactors, organic cofactors and the inorganic cofactors. In the given image, without the cofactors attached, protein is not active, but when the cofactors bindings activates the proteins. So, inorganic cofactors essential for proper activity of enzymes. For example, hexokinase has a cofactor magnesium, while the organic cofactors also necessary for the normal activity of the enzyme. For example, glycogen phosphorylase important for the proper organic molecule pyridoxal phosphate. In the given image as the enzymes cofactors, some enzymes requires cofactors to be effective, while the cofactors are a non-protein component of an enzymes, or the organic cofactors may be organic molecules. Inorganic ions cofactors may be permanently attached or the temporarily attached. The next one is about the types of organic cofactors. We have prosthetic group, it's a tightly bound organic cofactor, for example, flavin, heme groups, and biotin, while the coenzyme, it's a loosely bound organic factor, for example, NAD positive. As the prosthetic group and coenzyme. Prosthetic group with heme group, beta chain, and alpha chain, and helical shape of the polypeptide molecule, while the next one is the nicotine amide adenine dinucleotide or the NAD positive. Types of cofactors to be continued is that an enzyme with its cofactors removed is designated as apoenzyme, and the complete complex of a protein with all necessary and the small organic molecules, metalines, and other components is termed as the holoenzyme of holoprotein. We can see in the given image as the cofactors or the coenzymes, and apoenzymes becomes active by binding of coenzyme or cofactor to the enzymes, while the holoenzyme is formed when the associated cofactors or coenzymes binds to the enzyme's active sites. The next one is about the factors affecting the rate of enzymes. Factors that affect the rate of enzyme in catalyzed reactions are the temperature, hydrogen ion concentration, or the pH, substrate concentration. Now let's first talk about the temperature. How temperature affects the rate of the enzyme activity. For example, raising temperature increases the temperature of enzymes activity by increasing the kinetic energy of reacting molecules, while enzymes work best at optimum temperature when Q10 is the temperature coefficient, while Q10 equals to 2 for most of the biological processes. Sometimes heat energy exceeds the energy barrier, which may denature the enzymes. As in the given image, the temperature when it is between the 5 to 40 degrees centigrade, enzymes work best. When it exceeds the 40 degrees centigrade, it causes the denaturing of the enzyme. Now next start is about the effect of pH. All enzymatic activity also depends on pH. The optimal activity of enzymes ranges between 5 and 9 values. The denaturing of enzymes occurs if the pH is too low or too high, causes ionization. For example, in the given paragraph or in the given image is the increasing enzymatic activity when the pH is between the 5 and the 9, while when it is too low or high causes the ionization. Now I would like to talk about the Michaelis-Menten model and effects of substrate concentration. According to this model, the enzymes reversibly combines with substrate and yields the product and free enzymes. For example, in the given equation, E plus S gives the ES gives E plus E. What does it stands for? Well, E stands for enzymes and ES stands for enzyme substrate concentration and P stands for the products, while the K1 and K2 are the enzyme constants. In the given image is rate of reaction, while rate increases directly with substrate concentration, and this dotted line shows the maximum reaction rate, and rate approaches to the maximum, the red line. The next one is about energy and laws of energy transformations. What is energy and why we need energy? Everything that a cell does requires energy, and living things can only use chemical energy stored in their food. It's the force that enables us to work. There are two laws of thermodynamics. First law of thermodynamics, also called the law of conservation of energy, because it states that energy can neither be created and nor be destroyed, it can only transform from first to another form. For example, a squirrel eats a net, then uses its energy to climb a tree, and the total amount of energy in the universe is conserved. The next one is the second law of thermodynamics. It states that all objects in the universe tend to become more disordered, and the total amount of disorder is continually erasing. The degree of this disorganization is termed as entropy. For example, natural gas is burnt in a stove, and energy in the balls of gas converted into light and heat energy. Hydrogen energy used to boil water, and energy is lost in kitchen, so entropy increases. The next one is about the activation energy. Why the enzymes needs activation energy even they are very efficient? So let's see here. Minimum amount of energy required to start a chemical reaction is called the activation energy. Two types of reactions we have. The first one is the exergonic reaction, in which net release of energy takes place. The reactants contains more energy than the product. Well, this excess of energy is called the free energy, and these reactions occur spontaneously and exergonic reactions. Well, what are the endergonic reactions? Reactions in which energy absorbed. They don't occur spontaneously. The product contains more energy, so that's why they are called to be endergonic. As in this image, as the reactants, in terms of the heat of reaction and the activation energy converted into the products. The next one is about the ATP and how cells convert energy. As we all know that energy, ATP also called as the energy currency of the cell, and animal cells makes ATP in two ways: substrate level phosphorylation and chemiosmosis. So substrate level phosphorylation, ATP formation during reaction of glycolysis and Krebs cycle. ATP formation from ADP and P requires energy, and the reaction is coupled with an exergonic reaction. Enzymes transfers a phosphate group to ADP from the substrates. So now ATP is formed. Here we have the image of the substrate level phosphorylation, and enzymes transfers phosphate from substrate to ADP, forming the ATP and the product. Example is taken from the glycolysis in the cellular respiration. The next one is about a chemiosmosis, the coupled reactions in which the synthesis of ATP molecules takes during the movement of the hydrogen across the hydrogen gradient is called the chemiosmosis. The mechanism is given below. How the chemiosmosis takes place. The mitochondrial membranes have transmembrane channels, and these channels can pump proton. The flow of electron causes change in shape of protein. Proton moves out of the inner compartment of the mitochondrial, so proton concentration in the outer compartment of the mitochondrial becomes greater than inside. ATP synthase controls the synthesis of ATP during this process. And this we have a portion of the inner mitochondrial matrix, that is the intermembrane space. ATP formation takes place in the presence of the ATP synthase. Brief description of ATP synthesis is through the process of cellular respiration, glucose and oxygen converted into carbon dioxide and water, and energy transferred to ATP. An overview of ATP synthesis is: the first step is electrons are harvested and carried to a transport system, and the second step is electrons provide energy to pump protons across the membrane. The third one is oxygen joins with protons to form water, and the fourth one is protons diffuse back in driving the synthesis of ATP. Now I've done my presentation, and I wanted to close my presentation with my favorite quotation: is the secret of getting ahead is getting started. I hope so you are all enjoyed well my presentation. So give me thumbs up, likes and comments. Thank you.